Have you ever really thought about the very, very beginning of life? I mean, not just the basics, but the actual nuts and bolts biological architecture. Well, let's just dive right in, because it's an incredible journey. We're talking about the first 4 weeks of human development, where a single cell, all by itself, basically orchestrates the creation of an entire body plan, right from scratch.
You know, it really is the ultimate construction project, isn't it? It all boils down to this one question. How on earth does a single microscopic cell have all the instructions, all the machinery it needs to multiply, to specialize, and then to organize itself into, well, us? That is exactly what we're going to break down. So to get there, we're going to look at what I like to call the body's two photocopiers. Then we'll see how the essential ingredients, the sperm and eggs, are actually made. After that, we'll watch as this amazing 3-layered blueprint gets laid down, and then how that flat plan folds almost like origami into a body. And finally, we'll touch on what can happen when there are, errors in that blueprint. Okay, first things first. Before you can build anything, you need more building blocks, right? You need more cells. And our bodies have two totally different ways of doing this, each with a very specific job. Let's get to know them. Mitosis and meiosis. This really lays it all out. Think of mitosis as your standard high-quality photocopier. Its whole purpose is growth and repair. So it takes one cell and makes two absolutely identical copies. The chromosome number stays exactly the same. But meiosis is a completely different animal. It's a genetic shuffler. Its only job is to create sex cells, sperm and eggs. It ends up making 4 unique cells and, this is key, it cuts the number of chromosomes in half. Mitosis is this beautifully orderly dance. A good way to remember it is IPMAT. So first, in interphase, the cell doubles up its chromosomes. Then in prophase, they get all condensed and thick. Metaphase is when they all line up right in the middle. In anaphase, they get pulled apart to opposite sides. And finally, telophase, that's when the cell splits into 2 perfect clones. This exact process, by the way, is happening in your body millions of times right now. Okay, now meiosis is where the real magic of genetic diversity happens. It starts out similar to mitosis, with 1 round of DNA duplication, but then it goes through 2 full rounds of cell division. And in that first round, something amazing happens called crossing over. The paired chromosomes literally get together and swap pieces of their genetic code. This shuffling is exactly why you and your siblings, who come from the same parents, can look so completely different. So, now that we know about the genetic shuffler, meiosis, let's see how it actually makes the 2 key ingredients for a new life, sperm and eggs. Now, the processes are related, sure, but their timelines, they could not be more different. So for males, making sperm, or spermatogenesis, kicks off at puberty, and it's basically a non-stop production line. It's this super efficient 64-day process. It begins with cell growth, then right around day 40, the all-important meiotic divisions happen, and it wraps up with a 24-day maturation phase where these little round cells totally transform into the streamlined swimmers we'd recognize. The journey of an egg, on the other hand, well, that's a whole different story. It doesn't start at puberty. No, it starts before a female is even born. All the potential eggs she will ever have are formed while she's a fetus. Then they just sit there and wait, with a few starting to mature each month once puberty hits, all the way until the supply runs out at menopause. Now, to really understand how wild this is, just look at the numbers. At its absolute peak, around the 7th month of fetal development, the ovaries hold a staggering 7 million potential follicles. 7 million! This is the absolute maximum supply for a whole lifetime, and it's set before the person is even born. But here's where the story takes a crazy turn. Even before birth, a huge natural reduction happens. By the time of birth, that number has already plummeted from 7 million down to around 200,000. And what's even more incredible is that every single one of those potential eggs is paused, frozen in time, right in the middle of the first phase of meiosis, just waiting for a signal that might not come for another 15, 20, or even 30 years. And the decline continues. By the time puberty rolls around, only about 40,000 follicles are left. And out of that number, only a tiny, tiny fraction, we're talking maybe 400 to 450 in a lifetime, will ever fully mature and be released. It is just this incredible process of selection that starts long before life even has a chance to begin. Okay, so we've got our ingredients. After fertilization, that single cell starts dividing and becomes a ball of identical cells. But
how does that ball of cells know what to do? The moment everything changes comes in week 3. That's when the embryo organizes itself into a fundamental 3-layered blueprint. This massive organizational event is called gastrulation, and you could argue it's one of the single most important events of our entire lives. It's the process that takes a simple 2-layered disk of cells and transforms it into a complex 3-layered structure that basically sets the stage for every single organ and tissue in our bodies. So how does it actually work? Well, first, a little groove called the primitive streak shows up on the top layer of cells. You can think of this streak as a doorway. Cells from that top layer start moving toward it, and then they just dive right in. The very first cells to dive down form the new bottom layer, called the endoderm. The next wave of cells forms a brand new middle layer, the mesoderm. And all the cells that just stayed on top, they become the ectoderm. So, get this, that one original top layer gives rise to all 3. And this right here maps out what those layers are destined to become. The outer ectoderm, that's going to form our skin, but also, amazingly, our entire nervous system, the brain and the spinal cord. The middle mesoderm is the workhorse. It forms our muscles, our bones, our heart. And that inner endoderm creates the lining of all our internal tubes, like our digestive tract and our lungs. This is the master blueprint really taking shape. All right, so now we have a flat 3-layered blueprint. But obviously, we aren't flat creatures. So the next step, which happens in the 4th week, is this really dramatic transformation where the whole disc just folds and curves in on itself to create a recognizable 3-dimensional body shape. And this is not some random crumpling. It's a highly coordinated, beautiful process. The embryo folds in 2 directions at the same time. It folds from head to tail, and it folds from side to side, with the edges coming down and toward each other. And this whole movement is driven by the unbelievably rapid growth of certain parts, especially the developing brain and spinal cord. Honestly, the best way to picture this is as biological origami. This flat sheet of cells is meticulously folded and tucked, and it transforms into a C-shaped cylinder that starts to look, for the very first time, like a tiny little vertebrate. This folding is literally what builds the body. It brings the heart into the chest and pinches off the gut tube from the yolk sac. So, as you can probably tell, these first 4 weeks are just a cascade of incredibly precise, incredibly fragile events. The instructions in the blueprint have to be followed perfectly, which is why it's also important to understand what happens when they're not. So, what are the consequences when this intricate choreography has a misstep? I mean, errors can pop up at any stage, from that initial shuffling of genes way back in meiosis to the complex cell migrations of gastrulation. So for instance, if chromosomes don't separate correctly during that genetic shuffle of meiosis, it can lead to chromosomal abnormalities. And errors during the creation of the blueprint can have really profound effects. If that primitive streak doesn't disappear when it's supposed to, its cells can just keep dividing and form a type of tumor. And on the flip side, if there isn't enough cell migration at the tail end, it can cause the lower limbs to fuse together, a very rare condition called cyanomelia, or mermaid syndrome. So when you think about it, from the genetic shuffle of meiosis, to the cellular migration of gastrulation, to the physical folding of the embryo, this first month of development is just an astonishing sequence of events. It takes us from a single cell to a complex, 3-dimensional being that has a complete body plan. It really does leave you wondering, isn't this the ultimate biological choreography?